Okay, so today's video is going to focus on the cell membrane and cell transport. So we're going to first look at the structure and function of the cell membrane so we can get a feel of exactly what it's made of, which will dictate how it's going to work. So on the left, you can see your picture here of a cell membrane. Remember, all cells have a cell membrane. It doesn't matter if they're prokaryotic or eukaryotic. It doesn't matter if they have a cell wall. All of them have a cell membrane. And also remember, the job of the cell membrane is to maintain homeostasis. And remember, homeostasis is not just balance. Homeostasis is helping maintain that constant internal environment, trying to keep things the same internally all the time. And it's going to help maintain homeostasis via regulation. And by regulation, we mean it controls what goes in and out of the cell. And so the cell membrane, by what it's made up of, is going to control what's allowed to come into the cell and what's allowed to go out, which is going to help regulate the homeostasis of the cell. And its ability to do that makes the cell membrane what we call semi-permeable. And by it being semi-permeable, semi is half, permeable means that it lets things through. And so by being semi-permeable, it only lets some things through. All things are not allowed, all molecules are not allowed to cross the cell membrane. Okay? Only some of them are, and we'll talk more specifically about which ones in just a second. Okay, so let's look a little closer at the structure of our cell membrane, because that's what's going to dictate which of these molecules are allowed to pass through and which are not. Okay, and so the basic structure of our cell membrane is going to consist of these phospholipids. Okay, and that's all of these guys here. All of those little guys that look like they have tails, each one of those is an individual phospholipid. Okay, and you notice there's two of them, right? One is upside down and one is right side up. Okay, so these are my phospholipids, and they make up the majority of the cell membrane. They are arranged into what's called a bilayer. So there's two layers of them. We've got a top and a bottom layer of them. And each phospholipid has two parts to it. Okay? It has a phosphate group, okay, which is this circular part here. Okay? So that's the phosphate group. And that has a charge. And by a charge, we mean it's has a positive or a negative. That's what we mean by a charge. So it has a charge. Okay, it's not neutral. And then they have these tails right here. Okay, those little tails that hang off of them. Okay, and these are our fatty acid tails. And these are neutral. Okay, they do not have a charge. And in biology, when we talk about something having a charge versus not having a charge, we are talking about whether something is polar versus non-polar. And we're going to talk about polarity a lot when we do plants, but just so we can get used to those words, if it's polar, it has a charge, and if it is non-polar, there is no charge. And so these phospholipid molecules have both an area that has a charge as well as an area that does not. They have a polar and a non-polar region. And that's really important for its ability to regulate what is allowed to go through there. You'll notice with our bilayer, that middle layer is our neutral, non-charged, non-polar area. So all of this right here is non-polar. So our area of no charge is much larger than our area of charge. And so this area of no charge here actually wins out. So for example, molecules that have a charge, that are large, they are not able to get through the membrane. For a molecule to be able to pass through the membrane, it needs to have no charge. It needs to be small. Okay, those are the two biggest criteria there, that it needs to be small and it needs to not have a charge. And that's what allows it to slip right on through in or out of the cell. Okay? If it's too large, it can't fit between the phospholipids. If it has too big of a charge, this middle nonpolar area will kick it right back out. Okay, so it'll come in and it'll get kicked back out. So it will not be allowed to get all the way across the membrane. In addition to the phospholipids that are making up the majority of the cell membrane, we've got some other molecules that are also embedded there in the cell membrane. Okay, the first one that we're going to talk about are, see these little yellow things embedded in there? Okay, that is our cholesterol. Okay, and so what cholesterol does is it makes the membrane fluid. And by fluid, we don't mean that it makes the membrane into water. Okay, it makes the membrane flexible. It makes it able to bend and to change and to be flexible and not rigid. Okay, so cholesterol helps with that. It adds the spacing in there that adds to the fluidity of the membrane. Okay, you also may have some carbohydrates attached to these glycoproteins. Do you see the large green? So these green things that are sticking up there, you'll see that's our carbohydrate. And it's attached to what's called a glycoprotein. Okay? And so that glycoprotein, that acts almost as a name tag for the cell. Okay? It's the identification of the cell. It allows the cells to be able to recognize one another. So it allows for cell recognition. That's how your body knows that your cells belong to you, is what carbohydrates are attached on that glycoprotein. We also have integral proteins. Okay? These span the entire length of the cell membrane, so you notice it goes all the way across. Some of them help anchor in they anchor into the cytoskeleton, and so they help hold things in place. Okay? Some of them act as channel proteins, and so they allow molecules to pass through. Remember, in the phospholipid bilayer, we have that large area that is, so we've got this large nonpolar area in the middle there, right? And we've got some molecules that, remember, if they have a charge or if they're too big, they can't get through that area. And so we have what are called channel proteins. I think they look kind of like hot dog buns. And they span the length of that cell membrane, and so there'd be more phospholipids on the far side of it. And it spans the length of that cell membrane, and now molecules can go through this channel protein, and they never come in contact with that nonpolar region of the cell membrane. So your channel proteins are going to be very important for transport of molecules across that cell membrane. And so hopefully you can see how the structure of the cell membrane, the fact that it's made of this phospholipid bilayer that has both polar and nonpolar parts, so only molecules that have no charge and are very small can fit through it. But it also has these proteins that allow molecules to go through them, how this can help regulate what goes in and out of the cell. And so by doing that, it helps maintain homeostasis. So the rest of this, we're going to talk about how things get in and out of the cell. We're going to talk about passive transport and how things move with their concentration gradient. Okay, so passive transport is also called diffusion. Okay, so passive transport and diffusion are exactly the same thing. Passive transport requires no energy from the cell. Our usable form of energy, remember, is ATP. So passive transport is not going to require the production of any ATP. So if you have a cell that's doing a lot of passive transport, they wouldn't have the need, for example, for a lot of mitochondria. Remember, mitochondria make a lot of ATP, okay, and they would not require ATP to be able to transport things. With passive transport or diffusion, molecules also move from a high to a low concentration gradient. And you can see that down here in this picture. So in this picture, I've got my cell membrane right here through the middle. Okay? And so I have a high concentration of molecules on the outside and a very low concentration on the inside here. And so over time, So as time is increasing, these molecules slowly start to diffuse across the membrane from a high concentration to a low concentration. Think of it almost like going downhill. This doesn't require any energy for it to go from a high to a low concentration. And it's going to do this, okay, until it hopefully reaches what's called dynamic, that's an N, equilibrium. And dynamic equilibrium is you're going to have equal concentrations on both sides. Okay, so I've got equal concentrations on both sides, but once I get those equal concentrations, those molecules didn't just freeze and stop moving. What's happening is they're still going back and forth across the membrane, so that's our dynamic part, but just they're now going back and forth across the membrane at the same rate. Okay, and so we're at dynamic, there's the movement, equilibrium. So they're going back and forth across the membrane at the same rate. But this is our goal or our purpose for doing diffusion or passive transport. It's to reach dynamic equilibrium. And to do that, again, it does not require any energy. These molecules move from a high to a low concentration, and they do what we say, they move with the concentration gradient. Think of the concentration gradient like a river. They're moving with the flow. So now we're going to look at a particular type of diffusion or passive transport, okay, called osmosis. So osmosis is the diffusion of water. So if it's the diffusion of water, it's not going to require any energy. So there's no ATP that's required here. Okay, so no energy because it's diffusion, which is naturally does not require any energy. It's going to be the movement of water from a high concentration to a low concentration. Remember, diffusion or passive transport in general is the movement of something from a high concentration to a low. So in this case, we're specific for the diffusion of water. So it's moving water from a high concentration of water to a low concentration of water. And so that water is going to move with the concentration gradient. Okay, remember, it has the exact same properties of diffusion, just the only thing that's able to move right now is the actual water. Okay, so if we look at this, look at what we have up here. This is actually called a U-tube. And you'll notice if you look at the U-tube on the left, okay, they're at the same level. Okay, so the solutions are at the same level here, but one has more stuff in it than the other. Remember that stuff is called solute. When you have a solution, it has two parts, the solute and the solvent, and the stuff that's dissolved in the solution is the solute. So you have a lot more over here of all these little solute particles than you do on the left side. And so I have more water over here. I have a higher concentration of water on the left. And so that water is going to diffuse to the right. So I'm going to end up with a higher volume over here because my water went to the right to even out my concentration. You can see this down in the beaker happening as well. Okay, I have a higher concentration of water on the left than I do on the right. There's a lot more solute on the right. If there's more stuff in the solution, then there is less water. And so the water is going to move to that side. I'm going to raise my volume, lower my volume on the left, raise my volume on the right, but I'm going to end up at equilibrium. Again, it's a kind of diffusion, so my goal is to reach equilibrium. So I'm going to end up at equilibrium with the same concentration on both sides of the membrane. So molecules will do osmosis as opposed to regular diffusion because of that semi-permeable membrane. Remember we said in the beginning that the cell membrane is semi-permeable, which means only some things are able to get through it. So if we look at this membrane here, you've got these large solute particles here, and you've got these little tiny holes. So they don't fit, but the water does, which is why in this U-tube here, the water is what's going to move to the right, because that's the only thing that is able to. Yes, it might be easier to move the solute, but the solute doesn't fit because the membrane is semi-permeable. And we're going to do calculations with this. We're going to be able to calculate percent and which way water will move, and we'll practice with that kind of thing in class. When we're talking about osmosis, we're also going to refer to different types of solutions in regards to concentration. Okay? Hypotonic, hypertonic, isotonic. Okay? You can see I've got hypertonic, isotonic, and hypotonic. And so when we're referring to these, we're talking about the amount of solute. So that's what tonic is referring to, okay, is the amount of solute. So if something is hypertonic, then it has more solute than something else. If something's hypotonic, it has less solute. And if it's isotonic, it has the same amount of solute as something else. So let's look at what we would consider something being hypertonic. And so this is being applied to animal cells. You cannot apply this to plant cells because plant cells have a cell wall. And so they're not going to change shape like an animal cell will. So these animal cells here are in what's called a hypertonic solution. So let's put it in the solution. See these animal cells on the left that are all shrunken? So here's my beaker, and it's got some solution in it. And it's got this animal cell in it. And let's put some numbers with it because I think that helps people. Okay, let's say my solution is 90% solute. And my animal cell is 60% solute. So that makes my solution hypertonic. Hypertonic, hypotonic, and isotonic are relative terms. If my solution is hypertonic, then my cell has to be hypotonic. Okay, and that would make sense, hopefully, right? 90 is bigger than 60. Tonic refers to the amount of solute. So if I have 90% solute, then my solution is hypertonic to my cell that only has 60% solute. Remember that cell membrane is selectively permeable. So water is what can leave, not the solute. Water can move. So I need to take this information and I need to turn it into water concentrations. So if I have 90% solute, then that means I have to have 10% water. And if I have 60% solute, then I have 30% water in my cell. Water moves from high to low. So I have 30% water in my cell, 10% water in my solution. 30 is larger than 10, so water is going to leave the cell. And as water leaves the cell, as you can see up here, it will shrink up like that. And when it shrinks up, that's called crenation. So when an animal cell is placed in a solution that is hypertonic to the cell will lose water, so it will shrink or crenate, and it loses water, okay, because there is less water in the solution than there is in the cell, and water moves high to low. Let's look at a hypotonic solution. Okay, let's try a hypotonic. So now I've got a hypotonic solution. So let's say my solution has 70% solute. My cell has 80% solute. Tonic refers to the solution. So 80 is higher than 70. So my solution is hypotonic, which makes my cell hypertonic. I'm talking about water movement here, though. Water moves from high to low. I've got 30% water if I have 70% solute. So in my cell, I have 20% water. 30 is larger than 20, so in this case, water goes into the cell. Okay, and so water goes into the cell with a hypotonic solution. You can see that happening here. These cells are getting big and fat. They're swelling. And so when a cell is placed in a solution that's hypertonic to it, it will swell and can possibly go through what's called lysis, which means it can burst. Because remember, an animal cell does not have a cell wall, so it can explode, it can pop. And so these cells in a hypotonic solution can swell and pop or burst and go through lysis. So then an isotonic solution, if it's got the same amount of solute, so let's say we're at 70% solute, which means my cell then has 70% solute, which means I am at dynamic equilibrium. Water is not going to flood in or leave a lot, and the cell is going to stay the right size because my solute concentrations are the same. And if my solute concentrations are the same, then my water concentrations have to be the same. So we'll be working a lot with hypertonic, hypotonic, and isotonic solutions in class. So you can see how this would apply to a plant cell. It's a little bit different, again, because the plant cell has that cell wall. So you can see, again, I'm in a hypertonic solution, which means my solution on the outside here is hypertonic, which means my cell is hypotonic. And so let's put some numbers with it. I think that makes it easier. Okay, my solution here is 80% solute. Let's say my cell is 60% solute. Okay, so if I'm at 80% solute, then I have to be at 20% water. If I'm at 60% solute, I have to be at 40% water. Water flows high to low. So as you can see, water is going to leave this cell. But because the cell has a cell wall, it's not necessarily going to shrink the whole cell. It's going to shrink the cell membrane, though. You can see that's what all this is where it shrinks the cell membrane. And that's called plasmolysis. Okay, is when the cell membrane tears off of the cell wall. And that will kill the plant. That's how you kill a plant from not watering it, is when the plant goes through plasmolysis, it's been plasmolized, and the cell membrane has been ripped off of the cell wall. In an isotonic solution with a plant cell, so now my solution is equal. So let's say my solution is 60% solute, so my cell is 60% solute, which puts me at 40% water, 40% water. So my cell's at dynamic equilibrium, but it is what is called flaccid. Okay, and that's when a plant looks wilty and it needs a little bit of water because it doesn't have enough what's called turgor pressure. And that's going to be the last one we're going to look at, okay, is how turgor pressure affects it. Turgor pressure is the pressure on the inside of the plant cell. Okay, so this is the pressure that the contents of the plant cell are pushing on the cell wall. Think about if you blow up a water balloon, the more water you put inside the balloon, the more the water pressure is pushing on the sides of the balloon. Okay, that's how a plant cell works. And so a plant cell placed in a hypotonic solution, so let's say my solution now is, I like water to be blue. Okay, so, okay. So let's go with, let's make so we don't have a choice here. Okay, so we've got 50% solute in our solution, okay, which means I'm going to have 50% water. Okay, so I've got 70% solute in my cell, which means I have 30% water in my cell. So the water, remember water goes high to low, so water from 50 to 30, 50 is higher than 30, water is going into the cell. So the cell is filling with water. If you remember, in a plant and an animal cell that was placed in a hypotonic solution, the cell could swell up and explode because it doesn't have a cell wall. In this case, the cell will swell up, but it can't explode because it's pushing on this cell wall. And when it does that, the cell is said to be turgid. And that's actually when a plant cell looks like a healthy plant. That's what makes a plant stand up strong and tall without having a skeleton, is by having this turgor pressure. And so the last thing we want to look at with diffusion and passive transport is facilitated diffusion. This is a type of diffusion, so it is going to require no energy, no ATP, because it's a kind of diffusion. Okay? It's going to go from a high to low concentration again, because again, it's a kind of diffusion. So a high to a low concentration. So it's going with the concentration gradient. The difference here is that this is called facilitated diffusion. You know, when you facilitate something, you help it. And so facilitated diffusion is when they need proteins to help them, either a carrier protein or a protein channel. You'll see these green molecules are still moving from high to a low concentration. They just need help. And why would a molecule need help? Remember, it's either too big or it has a charge. It's polar. And so it can't slip through that cell membrane. And so it needs some help to get across that cell membrane. And so it will use either the carrier protein or, that's an R on the end, polar. It will use the carrier protein or it will use the protein channel. But it will use those to get across the cell membrane. So it still is going polar. Remember, polar, it has a charge. There we go, room to write it. So the molecules are too big or they have a charge or a polar. And so they use these protein channels or these carrier proteins to get from a high concentration to a low concentration. But again, that still does not require energy. Remember I told you to think about it like a river before? So if passive diffusion, you're going down the river, that doesn't require any energy to go down the river. But you know, some people can't float, and so they can't float down the river. They can, but if they had a raft or an inner tube, it still wouldn't require any energy for them to get down the river, but they could get that little bit of help. That's facilitated diffusion. Those people that sink to the bottom and they need a raft or they need an inner tube so that they can still float. So once they are floating, they are passive and they don't require any energy. All right, we'll look at all of this stuff in class. We'll do some labs with this. Okay, and we will also look at active transport on our next video, transport that's going to require some energy.